Hello! Welcome back to those of you who are following me. I have reached the portion of our AMP1 lab course where we are discussing cell structure and function, understanding the anatomy of the cell, as well as moving through the topic of the cell cycle and cell division, specifically the mitotic phase alongside G1S and G2, which we'll be describing. So first, let's begin very general with understanding the structures that are involved in maintaining homeostasis and normal function of a cell. First topic is identifying the portions and organelles of the cell and all the functions of these organelles that will make the cell functional and healthy. So the first thing that you want to look at in an animal cell, and that includes our cells, is that we, unlike very simple bacteria, have an enclosed nuclear envelope in which we have our DNA enclosed in this whole space called the nucleoplasm. So the entire structure is called the nucleus, and the space inside is called the nucleoplasm. So I'm going to start from the inside and work my way out. So in this nucleoplasm, we have the DNA material, the genetic material. And the DNA is wrapped up in these histone proteins, coiled up, and the structure is called chromatin, and it includes the DNA double helix all wrapped up in those histone proteins, and it decondenses during the cell cycle, which we'll discuss very soon. So the genetic code are sequences of nucleotides. So a sequence of 3 nucleotides makes up what's called a codon, which is the genetic code. And so those codons are located on the DNA double helix. Okay, so that's your DNA in here. Now, what about the nucleolus? So do not confuse the nucleus with nucleolus, which you can see in the center. So this is the ribosome making factory, and ribosomes, we're going to very soon learn that ribosomes assist in protein synthesis, and they can be found on this membranous structure called the rough endoplasmic reticulum, which is involved with synthesis of protein. Okay, now without any labels here, this structure here seen in green is called the Golgi Apparatus. And this is typically where the proteins are packaged and delivered. So protein synthesis occurs in the rough endoplasmic reticulum through the translation process of protein synthesis. There are 2 phases. Phase 1 is transcription, which occurs in the nucleoplasm, where the messenger RNA will transcribe and read the DNA codes. And then translation occurs where we are making a polypeptide chain, which are basically a chain of amino acids linked together. And so it's not a complete protein until it gets to this little green structure here. Of course, we color-coded it so you can see the difference between the Golgi apparatus and the endoplasmic reticulum. So the Golgi apparatus will take the polypeptide chain that was made in the endoplasmic reticulum, specifically the rough endoplasmic reticulum. And here is the Golgi apparatus, which will package and fold it up in a quaternary shape. So there's primary, where it's just a link of polypeptide chains attached to amino acids. It can be pleated, where it's folded. And then tertiary, it's folded even more, and quaternary is even more. So tertiary is 3-dimensionally folded. Quaternary is so folded up, it's like a almost like a ball and a thread on a yarn kind of thing. So those proteins are packaged here. That's the best way that I can explain it. So the cell has made the proteins, but they have to be folded up because they are very large structures and they take up a lot of surface area. So once they're folded up, then they can be transported. So they can be used by the cell or transported out. So these little tiny green balls represent the Golgi vesicles, and vesicles usually secrete or excrete contents. So what you see here, where my arrow is that Golgi vesicle extruding out the proteins that were made via a process called exocytosis. The vesicle, the Golgi vesicle, okay, is going to fuse with the cell membrane, and it'll invaginate and then release the proteins. And this process is called exocytosis, and it requires ATP, which is adenosine triphosphate. Adenosine triphosphate is a form of energy that is used by the cell. And speaking of ATP or adenosine triphosphate, who makes this energy? The mitochondria, which, if you've taken high school biology or in junior high school, you learned that the term, it's a coined term that everyone has heard. It's called the powerhouse of the cell. So the mitochondria is the powerhouse of the cell, and you can see it here. Now, you'll also see what's called cytoskeletal elements, and seen here, they look kind of like spaghetti strips together or cylindrical structures. These are called the centrioles, and they are typically, I don't know if you notice it here, but they're located 90 degrees to each other. And basically, it's composed of microtubules, which is a specific structure that assists and directs in movement, specifically the spindle formation, which is a structure that helps move the chromosomes during mitosis. And so these are all the structures that I can mention just over here in this image, but there's more to come. But wait, there's way more, so let's keep going. All right, so in this image, whoops, hold on a second. I'm having trouble with moving this. Right there. Okay, let me move my image over here. And my little thumbnail is over here, so let's keep going. All right, so some cells will have what's called, I'm going to work my way up now from the outside inside, whereas before we went inside outside. So let me move the thumbnail because it's still in the way. Okay, so let's start right here. You can see that you have the microvillus. And the microvillus, or microvilli for plural, these are located in digestive cells. So these aren't going to be located everywhere. Okay, so just so you know, this is like a cell that kind of shows all the possible components. Okay, so make sure that you know exactly what you're looking at, because you know, when we test you, we're going to test you on everything, not just one thing. Okay, what I'm trying to do now is move the center of this a little bit. Give me 1 second. It's very annoying because it's not always working to my, there we go. Moving it down. There. Okay. So I noticed that the basement membrane of this cell was not seen within the image. Okay, now let's continue. What you see here, you see these proteins being released. These are secretory vesicles, specifically the Golgi vesicles. And so if it's a Golgi vesicle, it's going to be secreting the protein. Now, seen here in yellow, whenever you see yellow, just think fat. Fat in the body tends to be like a creamy yellow color, and yes, you will see it there on a evaluation, identify cell structure and function. So you should be able to identify a fat droplet and compare it to the Golgi apparatus. The Golgi apparatus is granular. You could see the little tiny proteins in there. Okay, now additionally what you can see here, these little crista, these are the infoldings inside of the mitochondria, and this is where ATP is made. Okay, now here's the famous centrioles that I was talking about, which assist in movement of the chromosomes by creating the spindles, so spindle formation, and allowing the chromosomes to move during mitosis. We will get to that section as we move along. Now, over here we have the Golgi apparatus, and as I said earlier, this is where proteins are packaged and delivered. So I like to compare this to the post office or a FedEx store, right? Because you are going to have a product that you want to deliver to someone, and it has to be packaged and then it has to be delivered. So the post office and FedEx stores will do that for you. So that's the analogy. The Golgi apparatus is where we package and deliver via the trucks, and the trucks could be considered the Golgi vesicles, right? All right, and now let's go back to what we discussed earlier in the previous image. Here is the nucleolus and what's in the nucleolus. So here's the definition. I wrote it here on the side. Basically, the nucleolus is the site where ribosomes are made. And then the area here, the space seen here in purple, that is the nucleoplasm. And that's where our chromatin material is. And the chromatin is a combination of the DNA folded up in what's called histone proteins, Histone proteins. Okay, now the membrane around the nucleus is called the nuclear envelope, and these little tiny holes are called nuclear pores. And the reason for those are the fact that when the messenger RNA, it's a specific, very, very large structure, molecular structure, that will read the DNA code, and then it has to leave so that translation can occur and the protein can be made. So how does it leave? It leaves via these nucleopores. And so the messenger RNA will eventually attach to the rough endoplasmic reticulum, where there is a ribosome, and the ribosome is made of two subunits which will connect to the messenger RNA, and then each code is read, 3 nucleotides at a time, and each code represents a genetic code, and obviously another RNA has to come in with a specific amino acid for that code. And so that's what tRNA's job is, and we're going to go over that in a minute. So tRNA will come in and release that particular amino acid that matches the code with the messenger RNA codon, which is just a sequence of 3 nucleotide bases. How does it even recognize that? Well, because there's complementary nucleotide pairing, so that the tRNA has an anticodon, which is the complementary code or nucleotide sequence to the codon, which has the nucleotide sequence that's received and transcribed from the DNA. Okay, so I know it's a mouthful, and I'm going to keep repeating it as I go through this lesson. But that's pretty much the job of the rough endoplasmic reticulum. Now, smooth endoplasmic reticulum does not have the tiny little balls which represent the ribosomes. You can see them here, very tiny little red balls. But here it's smooth, and the smooth endoplasmic reticulum is involved in lipid synthesis, meaning making fats, lipid breakdown, and drug detox. Okay, so that's pretty much it. So please make sure you delineate that the smooth ER has no function in protein synthesis at all. Instead, it's the site of lipid synthesis, lipid metabolism or breakdown. Oh, and in addition, I forgot to mention steroid synthesis, and as I mentioned before, drug detoxification. Okay, so again, smooth ER, what is the difference between smooth ER and rough ER? This is typically a question that you will find over and over again in lecture and lab and across all anatomy and physiology courses. Most professors will ask for you to know the difference. It is quite important. So I'll delineate it one more time. The smooth endoplasmic reticulum is the site of steroid synthesis, lipid synthesis, and lipid metabolism, okay, and drug detox. All right? And I think that's all my notes on that for smooth. Now, rough endoplasmic reticulum, as I said, protein synthesis occurs there. We're talking about this area here, and it's the second part of protein synthesis, and it's called translation. Protein synthesis has two phases: transcription, which occurs in the nucleoplasm, and transcription involves the messenger RNA transcribing and reading the DNA code. And then that messenger RNA leaves the nuclear pore and attaches to a ribosome on the rough endoplasmic reticulum, and that's where that factory making center of creating the polypeptide chain of amino acids begins. And so we call that phase translation, where the mRNA attaches with the ribosome subunits, two subunits there, and then the tRNA, each and every single tRNA will come in and complementary bind with the codon. So the anticodon sequence, nucleotide sequence on the tRNA binds with the codon sequence on the mRNA, and each and every single time it binds, another amino acid is going to attach to the previous amino acid until each code is read and translated, and each amino acid is linked to the previous one, and you have a resulting polypeptide chain. And at the end of the whole translation process, there is usually a end codon, which is a stop codon, and that will release the polypeptide chain from the factory making center there, from the ribosome and the mRNA. So therefore that polypeptide chain goes where? It goes right here to the Golgi apparatus, okay? The mRNA goes back into the nucleoplasm for another job to do, and the ribosome hangs around for another session of protein synthesis. And that's how the cells constantly make proteins. And you probably are wondering, well, why do we need so many proteins? Well, if you think about it, our body needs a lot of proteins for a lot of the contents. So for example, the receptor sites on cell membranes are made of protein. The antibodies that kill bacteria, the antibodies that fight bacteria and viruses, okay, these antibodies are made of protein. Okay, so there are a lot of protein, even like specific enzymes that break down things in our body for our digestive process are also protein. Okay, that's why we need so much protein. All right, so now I'm going to keep going because I really spent a lot of time on that. So let's see over here. This next slide, let me see, maybe I'm trying to clear it, put it right dead center. I want to make sure you can see all of this image. There we go. And I'm going to move this. Oh, I'm going to move it right here. Okay, so in this image, what we are looking at is basically, I still feel like I'm half blue, half white. Let me move my thumbnail here. There. This image just shows the cell membrane. So if we describe the cell membrane, it has two layers of lipids, phospholipids specifically. And if you ever did the experiment of putting oil in water, you know that it doesn't mix. Okay? So it's hydrophobic. Fats. All right? So oils are fats. lipids. Okay, so lipids are hydrophobic. They do not mix with water. So by the nature of our body, outside extracellular fluid, we have lots of water. Intracellularly, we have lots of water. So obviously, the lipids don't like that, so they hang out together. So the lipid tails hang out together. All right? And so when that happens, the hydrophobic ends are binding or causing a very nice tight fit and boundary between the outside and the inside of the cell. So the analogy could be made is if I get a white bread sandwich, okay, or brown wheat, whatever you want to, whatever bread, and you put butter and you stick it together, right? It sticks because butter is fat and these are lipids, okay? So you get a nice sticky sandwich that won't spread very easily unless you pull it apart. So that's how our cell membrane is formed, okay? It's a double layer of phospholipid. That's the structure of the membrane. Now, in addition, I would like to add to the analogy, if you were to add olives with a toothpick and stick it into the sandwich, it would be not a very tasty sandwich, right? It would be a toasted, buttered bread with olive. So that would be the sandwich. Now, why am I even mentioning food? Well, because if you picture this as being an olive, it's dead right in the center. And so these are the receptor sites. And so these are channels that can open or close based on passive and active membrane transport, which we will be going over in a minute. Okay? So that's pretty much it. Now we have proteins as these channels that are integral, meaning integrated into the cell membrane, and then we also have peripheral proteins, which are not integrated within the cell membrane, but directly inside, right on the inside of that cell membrane. You will also see these chains, these glycoprotein chains. These are typically found in many cell membranes, depending on what also the protein channels and their structures are made up of. Okay, I'm going to keep going now. So in this image, we're going to see more of the same. We're going to see the plasma membrane. And let me move this over here. Okay. So this is a better view showing you how the peripheral proteins and their chains, their glycoprotein chains, or they could be any kind of carbohydrate chain, are sticking out. And here's the hydrophobic lipids. Here's a protein that spans the entire cell membrane, so we call that a transmembrane protein. And here's one with the channel that opens and closes depending on the membrane transport that we will just be discussing. Intracellular fluid, extracellular fluid, this is the lipid. Okay, so we were at the plasma membrane before I got interrupted. Let's continue. So we were talking about the different types of protein channels. So we have the transmembrane protein, the peripheral protein, and we also talked about the phospholipid bilayer and how it is hydrophobic. Okay, so the next topic I'm going to discuss is the structure of the cells. So there's a lot in here. I don't even know if I can fit all of it. Let's see. Let me do this. Oops, that's not what I want. All right. Here we go. All right, so the first thing I want to discuss is the nucleolus is where, as I mentioned before, the chromatin is. And then the the nucleoplasm is where the chromatin is located. The nucleolus is where we make ribosomes. And then we have the nuclear pore, which is the location in which the mRNA departs. And there's multiple nuclear pores. Okay, don't forget the chromatin is made up of, guess what? DNA plus the histone protein. Now in this image you can see a lot better the rough endoplasmic reticulum as opposed to the previous one I showed you. So here we see the rough endoplasmic reticulum with the ribosomes. This is the site of protein synthesis. And then right here you can see the smooth endoplasmic reticulum, which is the site of steroid synthesis, lipid synthesis, lipid metabolism, and drug detox. So 4 things. One thing for the rough endoplasmic reticulum, 4 things for the smooth endoplasmic reticulum. So you need to remember that. All right, what do you have to know about the nucleoplasm? In the nucleoplasm, we have the chromatin, which is your DNA double helix wrapped up in histone proteins. What do we have to know about the nucleolus? Inside is the factory making center where we're making ribosomes, which are going to assist in protein synthesis at the rough endoplasmic reticulum. What do we have to know about the Golgi apparatus? It is the factory making center to package and deliver those proteins that were synthesized in the rough endoplasmic reticulum. Okay, so the rough endoplasmic reticulum will make a long chain of amino acids. So it's a primitive, unfinished, yet to be developed protein. In other words, it's just the polypeptide chain of amino acids, which does make up a protein. But once the protein gets folded up, either, well, in this case, it'll be folded up quaternary. But if you learn about how proteins fold, which was in chemistry, it could be pleated, it could be tertiary, or quaternary. We're talking quaternary, where it's really folded up very nicely. And that's done in the Golgi apparatus. And then here you can see the Golgi vesicles fusing with the cell membrane and secreting the proteins from the cell via exocytosis, which is a form of active transport requiring ATP. And ATP is developed and synthesized in the mitochondria, and you can see the infoldings here. These are called crista. Also, in addition, we talked about the centrioles, which are located 90 degrees to each other, and the centrioles assist in the spindle formation. You can see spindle here, and the spindle is a structure kind of like a highway system in order for chromosomes to move. All right, certain cells will have microvilli, microfilament, microtubules. So these are all the structures that keep the integrity and allow for movement inside the cell, kind of like a highway system. Now, the last but not least, I want to discuss peroxisomes and lysosomes. So make sure you know the difference between a peroxisome and a lysosome. So here's the difference. The peroxisome is smaller, the lysosome is larger. I don't know if you can tell in this image, but it is. The peroxisome releases peroxide, and it's particularly abundant in liver and kidney cells, and basically it's there to detoxify alcohol, free radicals, and other harmful chemicals. So if you drink a lot or you're doing drugs, peroxisomes will develop more. The cell will start to evolve and adapt, basically, and build more peroxisomes to detoxify. Okay, so that's peroxisome. Now, what is a lysosome? So the lysosome is, as I said, slightly larger, and it contains digestive enzymes called acid hydrolases, and they function as the garbage truck, basically. They break down worn out cell parts. Okay, so that's how I remember it. Digest worn out cell organelles and foreign substances that enter the cell. We like to call them suicide sacs, okay? Because for obvious reasons, okay? All right, I think I mentioned everything in this image. The only thing I didn't talk about was the nuclear envelope. It is also 2 layers, as is the 2-layered cell membrane biphospholipid layer. Okay? All right, so let's take a peek over here, and we are going to look at the microvilli. So the microvilli are basically going to be found in definitely intestinal and reproductive areas like the uterus, for example. Microvilli are basically there to increase the surface area and allow increased absorption and movement of the molecules or whatever we're looking at. So these are the microvilli. They're finger-like projections. You're not going to find them in every single cell, but that's what they look like. Here you can see the mitochondria, the Golgi apparatus, the nucleolus where the ribosome is synthesized, the nucleus with the nucleoplasm. The nucleoplasm is the space inside, and of course the chromatin is in there. So you're going to see this as a universal structure where you're going to find all these things, all these structures, all these organelles. And don't forget the rough endoplasmic reticulum, the factory making center for protein. All right, so here I have just a small little chart that summarizes pretty much everything that I've just discussed. So the one thing I didn't talk about in detail, because I said, oh, the 2 subunits in translation when we're doing protein synthesis at the rough ER translation phase. I did not discuss the exact two subunits, but now you can see what the ribosome looks like. And so in the middle, in between these 2 subunits, would be the messenger RNA, this long strip of nucleotide sequences that was transcribed from the DNA. So the ribosome's main purpose is that they are tiny spherical bodies composed of RNA and protein. They like to float free, and they also will be attached to the rough ER in the cytoplasm. And so this is the actual site of translation phase of protein synthesis. Now the endoplasmic reticulum is a very interesting site, because I already told you that if it's the rough ER with the ribosomes, we're making protein. Okay, so it provides an area for storage and transport of protein that's made on the ribosomes. Okay, and then of course if we look at the smooth ER, which has no function in protein synthesis, instead is a site for steroid and lipid synthesis, lipid metabolism, and drugs, as mentioned before. Right, so now in this image we can see, let me move my thumbnail, we could really see blown up here the contents inside, which is the chromatin. So what was the chromatin again? The chromatin was DNA, all right, plus the DNA plus the histone protein. Okay, and then the nucleolus, we were talking about the fact that inside the nucleolus we are synthesizing ribosomes. And then we also discussed earlier, as you can see here with my arrow, the nuclear pore. So if the chromatin unravels, the DNA unravels from the histone protein and is one long strip, so basically the helix uncoils and a portion of the DNA is exposed. We call it the naked DNA strand. And that naked DNA strand is now exposed to the mRNA to transcribe it. Once it's transcribing the DNA, there's a stop codon, just like in translation. There's a stop codon in transcription, and therefore the mRNA is released from the DNA and departs via the nuclear pore. Okay, so more of the same. Please make sure that you study the structure, and you will be able to hopefully identify all the structures in the cell. Okay, so to quickly review one last time, don't forget about the mitochondria, the powerhouse of the cell. We are making ATP there. Don't forget about the smooth endoplasmic reticulum and its function. Don't forget about the rough endoplasmic reticulum and its function. So this is smooth, this is rough. Don't forget nucleolus, chromatin inside the nucleoplasm. Chromatin is wrapped up DNA in a histone. Okay, and don't forget the function of the nuclear pore, as well as the centrioles, which are 90 degrees to each other, forming the spindle, the packaging and delivery of proteins at the Golgi apparatus, etc., etc. So more of the same, you have the definitions here. Packaging proteins, Golgi apparatus. Lysosomes basically are digestive enzymes that have acid hydrolases, and they digest worn out cell parts, so they are suicide cells. Okay, here's the structure of the mitochondria, and as you can see here in bright red, we are making ATP. This is the factory making center for ATP, and the infoldings inside are called crista. Here you can see in this image the free ribosomes versus the membrane-bound ribosomes on the rough endoplasmic reticulum. In this image, table 4.1, you can see that you have a peroxisome, which has the membranous sacs containing the enzymes that detoxify. So don't confuse peroxisomes from lysosomes. Lysosomes are suicide cells. They digest worn out cell parts. Peroxisomes are a bit smaller than the lysosomes, and they detoxify alcohol, they have hydrogen peroxide, and they detox harmful chemicals. Mitochondria already I told you a million times, so you should know by now. Hopefully it'll stick with you. Powerhouse of the cell, we're making ATP. Alright, now here's the beautiful image here of the endoplasmic reticulum. Alright, let me move this over here. Ugh, come on. There. Okay, so here you can see the endoplasmic reticulum. We have smooth endoplasmic reticulum without any ribosomes, and then we have rough endoplasmic reticulum studded with little tiny ribosomes. And your nuclear envelope, you see how it's like a continuous maze with the rough end. So you're always going to see the rough endoplasmic reticulum really close to the nucleus, and it makes sense, right? Because part 1 of protein synthesis, transcription, occurs in the nucleoplasm, and part 2 of protein synthesis, which is called translation, occurs in the rough ER. So this is really a nice view how the rough versus the smooth looks in terms of the membranous structure. Alright, let me move my thumbnail. Again, if you would like to see the definitions, here they are. This is from McGraw-Hill, and I'm assuming you can look this up in your book, but I put it here just so you know the definitions, because you will be responsible for knowing the structures and the functions. Okay, so the location is in the cytoplasm. The centrioles are 90 degrees to each other, and formation of the mitotic spindle during cell division, it forms the bases of cilia and flagella. So flagella are the tail end of sperm, so that's the only place where you'll see flagella. And cilia you'll see in specialized cells in the body. Okay, cytoskeletal elements is the last structure that I would probably want to describe. There's 3 different types. Microfilaments are made of very large molecular structures that contain actin, which is a contractile protein, which we will discuss when we do muscle contraction, and is therefore a very important structure, molecular structure, in providing cell mobility, particularly in the muscle cells. The intermediate microfilaments right here. The intermediate filaments are stable elements composed of proteins and resist mechanical forces that are against the cell, acting on the cell. And then finally, the microtubules, which form the internal structure of the centrioles and help create and maintain the cell shape. All right, and in this image, we can see the Golgi apparatus, and alongside the Golgi apparatus, you can also see the transport vesicles. You'll see them in various books either as Golgi vesicles or secretory vesicles. Basically, they mean the same thing. Okay, so they kind of form from the actual membrane of the Golgi apparatus, and then they separate out and become these secretory vesicles or Golgi vesicles. And how does that happen? Well, once the proteins fill up inside these vesicles, then they are released and transported to the cell membrane for exocytosis. All right, so here's another image. This is a microscopic view. 600 nanometers, that's how it's electron microscope, and you can see that they do exist. We're not making this stuff up. This is the Golgi complex, okay, right there. All right, there's your lysosomes. And let me move my thumbnail here. So the lysosome structure, basically it has a glycosylated membrane, and it has the hydrolytic enzyme mixture inside. And believe it or not, it also has a lipid bilayer, just like the cell membrane. So that is the template. So our cell membrane holds everything inside the organelles, and then these organelles also have these bilayers. So this particular organelle, the lysosome, has a lipid bilayer as well. Okay, I'm going to keep going. What are we looking at here? In this image, we are looking at how a vesicle is fusing with the membrane. So let's start at 1. We have the nucleus here, and we have the invagination, and that's step 2. Phagosome, lysosome. So we engulf a structure here. It could be anything. It's a particle. It's a foreign body, and we don't like foreign bodies in a cell, right? So the cell will destroy it with the lysosome, and so that's what you're seeing here. The lysosome hydrolytic enzymes releasing and breaking it down, and this is the residue that it releases. Okay. All right, let's keep going to the next slide. All right, now we talked about, I can't move my thumbnail, the peroxisome. This is again the structure, lipid bilayer, and you can see exactly. Note that the peroxisome are the ones that are found in the liver. What else do I want to say? Yeah, liver and kidney cells, because they detoxify. It makes sense. Okay? Because the liver and the kidney, that's you. Okay, now this next slide shows you in a nice little organized fashion in this table all the functions of all the organelles inside the cell. So if you are studying this, you might want to put these on index cards. Perfect time. Screenshot this and take some notes. All right. Now moving to the centrioles. Let's move to the centrioles. I'm going to try to move this whole image. Hold on 1 second. There we go. And I'm going to move myself here. All right, so here's the centrioles. We have a centriole. They are located 90 degrees to each other. Now notice the term centrosome. Centrosome is the entire region which contains the centrioles that are 90 degrees to each other. And the centrioles are made up of microtubules. and the centrioles come as a pair, and like I said, the pair together make up this whole section called the centrosome. And the region, the centrosome with the centrioles, will form these spindles, and when it comes time for cell division, we will see the spindle form, and it allows the chromosomes, these are the chromosomes, to move. Okay, so let me move this portion down again. So make sure, so here's what I'm talking about. I'm going to move this here. The spindle pulls the chromosomes apart, and we will be going over this. When the chromosomes separate apart, that phase is called anaphase, and eventually we'll have two equal daughter cells. Okay, so that's that. So let's keep going. We're almost done. This is the cytoskeleton. And the cytoskeleton is composed of the filaments, which creates the shape and integrity of the cell, also like a highway system, because things don't just stay stationary in there. They move just slightly because of these structures. Okay, so let me move this down, and I'll come up this way. So basically, what you need to know is, what are the components of the cytoskeleton? These are the intermediate filaments, you need to know that, the microtubules, and of course the microfilament. Okay, microtubules are large, intermediate are kind of small, right? And then medium-sized cylindrical structures are the microfilaments. All right, here we see the cilia. Like I said, digestive, mucus, respiratory. You can see cilia in various cells. We will go over that. But this is, mucus forming, and the cilia can be found in our cells in the nose, and it's a way of cleanup. Think of it as a vacuum almost. It's just like the brush on the vacuum. All right, and then of course the flagella on the sperm. There's this cute little, isn't that cute? That is a stuffed animal, but it's really a stuffed sperm. Fabulous slide. Here it is. Okay, so what's the component of the sperm? Is that you have the acrosome, which has an enzyme that's going to, break through the egg. The nucleus, which has the DNA. You've got the centrioles here. You've got the midpiece, which contains the mitochondria, and here's the sperm having the flagella. So this is the flagella, the tail. Okay, so the end portion is filament, and it allows movement of the sperm. All right, so with that said, you have this PowerPoint, which I converted to a PDF file so that we can watch it in virtual classroom and discuss. But I encourage you to study and be able to practice on your own identifying the structures unlabeled. And you can find all different kinds of cells online. You can even Google some and just have practice. But your book has it. This document also has it. You can practice identifying the structures. So B, for example, is centriole, C is nucleolus, etc., etc. So I'm not going to go over that because we went in detail already. Okay, now the next topic at hand is very important. It is called the cell cycle, and without the cell cycle, how can I put it? The cell would not be alive. The cell needs to continuously replicate itself, and at some point it needs to stop replicating itself, and that term is called apoptosis. A-P-O-P-T-O-S-I-S. And apoptosis is cell suicide. If apoptosis doesn't occur, that's cancer, when cells keep multiplying and multiplying and multiplying, and the cells age, so after a certain amount of cycles it needs to die. All right, so let's go over phase 1, 2, 3, and 4. So phase 1 is called G1 or first gap phase. This is where a lot of growth and normal metabolic roles occur. This is the second phase is called S phase, and if you see the DNA, it's a double helix. It unravels itself. Okay, so now you have two single strands, and then you develop a double helix on this single strand and a double helix on this single strand. So now we have duplicates. And so we do that because the cell's going to divide. So that's the S phase. The G2 phase is more growth and preparation for mitosis. And then we go through the M phase, mitotic phase. So first phase is prophase, second phase is metaphase, third phase is anaphase, fourth phase is telophase, fifth phase is cytokinesis. And the way I remember it is P-MAT, P-M-A-T, P-MAT. So think of a puppy, peas on a P-MAT, P-MAT. Okay, so now we're going to go over each phase. So let's do it. So phase 1, let me move this over to the side over here. Okay, moving the thumbnail to the side, and we see the phases. Okay, so let's talk about most of the cell cycle will be interphase, because interphase is a very, very extended portion of the cell cycle. Interphase equals G1, S, and G2. I repeat, interphase involves G1 phase, S, and G2. Okay? And so when you look at interphase, you're going to see a very distinct nucleolus, a distinct nuclear membrane, and that's pretty much going to tell you that nothing is happening, but something is happening. It's the preparatory phase, but cell division, mitosis, is not happening. Now, when the cell starts the first phase of mitosis, early prophase, you will start to see the breakdown of the nuclear envelope. The nuclear envelope breaks down, and notice in interphase you saw the nucleolus. In early prophase, the nucleolus disappears. Okay? And the chromatin transforms into chromosome. Okay, so it condenses. So we have chromatin here, looks like spaghetti, or more like linguine. And then when it goes to this early prophase, the chromosomes will condense and become visible into two sister chromatids held together by the center, which is called a centromere. And don't confuse with centrosome and centriole. I get that a lot from students. Centromere, centrosome, centriole. Okay? So what's happening in early prophase? As I said, the nuclear envelope breaks down, the nucleolus disappears, and the chromosomes appear. Okay, then what? In late prophase, we no longer see the nuclear envelope. It is gone, and we definitely visibly see the chromosomes. The late prophase, you'll start to see the spindle forming. Okay, so what are the highlights? In prophase, early prophase, nucleolus disappears, nuclear envelope disappears, chromatin become chromosomes. Late prophase, the nuclear envelope is gone and the chromosomes are visible, spindle starts to form. Okay, let's go to the next slide, and this shows you interphase. So let me move this up now again, hold on. Give me 1 second, patience, patience. It's nerve-wracking. Where is it? There it is. I can't do it. Dang it. There we go. Maybe I can move it from here. There. It's the image in place. Let me try moving it here. Okay, well, whatever. I can't fix that. All right, let's show you this here. Maybe I can move it now. There. There we go. That's better. I apologize. I'm having technical difficulties. Okay, so interphase, as we said, there's no early prophase, nothing's happening. You can see the nucleolus, okay, and you can still see the nuclear envelope. The chromatin is located there. So that's interphase, okay? You're going to look for the nuclear envelope, and you'll know that it's there in interphase, and you'll also see the nucleolus. Okay, now over here, let me just make this a little bit smaller. There. Nope, that's too small. That's not what I wanted. Hold on. Okay. There. That's what I want. Ah, it did it again. Dang it. It won't let me do it. Alright, so let me do this again. Take two. Very annoying. Okay, I guess I'll have to deal with it this way. So now we're talking about early prophase and late prophase. What's the difference? And there have been issues with this when it came to taking tests, because it could be a little bit confusing, but if you take a look at more slides, I would really prefer that you look at plenty of microscopic slides and see the difference in appearance. If you look at early prophase, you could still see the remnant and boundary right here of a nuclear envelope, whereas when you look at late prophase, you really don't see that at all. Now, I could see the argument that you can't really tell, you know, these chromosomes from these, but look at the difference. This is encased around a membrane, and this is like a starburst appearance, and there's nothing surrounding them. So that's how you could tell early prophase from late prophase. All right, now the next phase, so we did prophase, the next phase is metaphase. This is really easy because the chromosomes line up at the equator or at the center of the cell, and these are called the spindles. So this, if you get on an exam, is a gift. It's so easy to identify, so make sure that you get this down cold. And this is called the metaphase plate or equator or center, right? And these are the spindles, and of course there's your centrioles, and the centrosome is the region. And don't forget, the structure holding the two sister chromatids together to make up a chromosome, again, the structure that's holding the two sister chromatids to make up a chromosome, that structure is called the centromere. Do not confuse centromere with centrosome and centriole, which are the 90-degree structure, cytoskeletal structures. Okay, next is anaphase. Ana gets a divorce. When you get a divorce, you go to opposite sides of the house. So this is what's happening. The chromosomes are being pulled by the spindle, and the chromosomes go to opposite polar ends of the cell. And what do you see here? But the next and final phase before cytokinesis, which is cell separation. So telophase is the last phase where the cell is now pinching in. We call that cleavage furrow. It's a contractile ring that forms. And you can see in the center, the chromosomes are kind of gathered together at the center. You could still see the spindles. All right. And after that, you'll see that the cytokinesis, let me see if I can find cytokinesis. Hold on. Where is it? I'm going to move ahead and then come back. It does not like when I'm moving it fast. Okay, well, we'll have to get to cytokinesis when we get to it. I don't see it. Okay, so once the cell completely breaks apart into two equal daughter cells, then it's cytokinesis. I know I have a slide on it somewhere. Okay, so to review again, telophase is where the cell pinches in, and that's called the cleavage furrow. All right, so this shows you a review of all of what we just discussed, from metaphase to anaphase to telophase and cytokinesis. All right, so metaphase, everything's at the middle. Anaphase, the chromosomes are pulled apart. Telophase, the cleavage furrow, and you can see that it separates into two daughter cells. All right, so make sure you learn this. Have it memorized. It should not be so hard. If you've done this in high school, this is easy peasy. So again, interphase is the phase of preparation, G1, S, G2, and then mitosis, early. Prophase, the nucleolus is still there, the nuclear envelope breaks down. Late prophase, the chromosomes become visible, the spindle forms. Metaphase, the chromosomes line up at the center. Anaphase, the chromosomes separate because of the spindles to opposite polar ends of the cell. Telophase, cleavage furrow, nuclear envelope reappears, the chromosomes become chromatin again, and then we've got pretty much a complete cell division into two equal daughter cells. The movement of separation is called cytokinesis, and the cycle continues. All right, so in this image, we see, as mentioned before, the centromere. Do not confuse with centrosome and centriole. And chromatid is one sister chromatid, and this is another chromatid. So they are complementary in terms of its DNA. All right, so let me review again. You've got metaphase, anaphase, telophase, cytokinesis. And what are we looking at here? You can see the nucleolus, you can see the nuclear membrane, and you can see the cell. So that's how you know that this cell is undergoing interphase, G1, S, or G2. It's just prepping, because there's its nuclear envelope. There you can see the chromatin, and that is where the nucleolus is. So that's interphase, okay? Prophase. You start to have the nuclear envelope breaking down. You see it's breaking down here, and the chromosomes become visible. See? And then the spindle forms in late prophase. Metaphase. Everything lines up at the center. Okay? And so that identify. This thing does not want to cooperate. Okay, so this is metaphase plate, showing you how the chromosomes are lined up. And then this is anaphase, showing you how the chromosomes separate. You can see here the spindle. And over here you can see the daughter cells, equal daughter cells, going to opposite polar ends of the cell. This is anaphase. Telophase, you can see the cleavage furrow forming here. The cell is pinching in. There's your chromosomes. And you can see how the spindles are still visible. And cytokinesis, there's the slide I was looking for. And my laptop was just not letting me move forward fast enough. Okay, so here it is, the contractile ring, and it's completely separating out the cells. All right, so cytokinesis refers to the pinching of the cell into two new daughter cells, which is accomplished by a narrowing contractile ring. All right, so in this table, you'll find in McGraw-Hill the chromatin condensed, so it just gives you all the steps in table format. I'm very visual, so I like to show you those first. So prophase, as we mentioned, I'm just going to highlight the chromatin condensed to become visible chromosomes. The nuclear envelope breaks down, the spindle starts to form, the nucleolus disappears. Metaphase is easy because all you have is the chromosomes lining up at the center. Anaphase is when the chromosomes go to opposite polar ends, they migrate to the poles. Cytokinesis often begins. Telophase is the chromosomes reach the poles, nuclear envelope reforms, right? The chromosomes unwind and become chromatin, and the cytokinesis starts to happen and divide the cytoplasm. The nucleolus, of course, reappears because it disappeared during the mitotic phase and separation. So now the nucleolus reappears and we move back into interphase, prep phase. Okay, now this next slide is going to show you whitefish blastula, so developing whitefish, and how you can have different stages of mitosis. So this is a histological slide. So that was prepped and dyed with a cover slip. You can look under the microscope and see the different phases. Okay, so this is anaphase, this is prophase, this is telophase. Don't worry, I'm not giving you small cells like this to look at. When you have your quiz and when you have your practical, you will have very large pathology slides as well as the cell models from the lab to be able to identify the phase of mitosis. All right, and this is the last slide showing you, believe it or not, this is an onion root tip, and it's a longitudinal section showing you this is metaphase right here, this is interphase, okay, and then if you look up over here you can see telophase because it's separating out. There's anaphase with the spindle forming and pulling the chromosomes to the opposite polar end. Okay, so sorry about some of the technical difficulties. I'm getting used to navigating with this Screencast-O-Matic and then uploading it on YouTube. So I do hope that a narrated PowerPoint is more helpful than you just reading and looking at this before our lecture and lab sessions. All right, so I'll see you week 2. Or in certain semesters it's week 3 based on holidays. So this is typically done at the beginning of all A&P courses. So whoever's watching this, I do hope it helped. Take care. Bye.